Good morning and welcome to day 3, final day of Devoxx US. Tons of interesting things on the schedule this morning. We have lots of language and platform and mobile and embedded stuff coming up. But first, we have 2 keynotes this morning. We're going to hear from Olivier Gevaert talking about biomedicine and TensorFlow. And then we have a talk on a slightly different topic. Brian Kane comes from the Rhode Island School of Design, and they started or have the only AI design program in the world. I would be willing to believe that. He's going to be talking about art, AI, and apparently wallfish. So enjoy the keynotes, and I'll see you in a few minutes. Oh, one important note. We do have a timing issue this morning. We're probably going to run a little bit long, which means that the first talk is going to start at 10 after the hour instead. So it's going to be 10:10 to 11, and then we're going to push all the morning talks by that same amount. So everything on your schedule, it says it starts at the hour and runs to 10:2. We're going to push that by 10 minutes. It starts at 10 after the hour and runs to the hour. Olivier, it is your show.
Good morning. So this morning I want to introduce you in how we are using deep learning in biomedicine. So my name is Olivier Gevaert, and I'm an assistant professor at Stanford University, and we essentially investigate how we can help cancer patients to develop treatments that are better focused on their tumor instead of general populations. So deep learning and software implementations such as TensorFlow have a huge potential in biomedicine. However, not all biomedical data is immediately suitable for deep learning, so there's a lot of challenges. And the biggest impact is what is now called precision medicine. And what is precision medicine? So precision medicine is essentially treatment of patients where we really tailor the therapy to their disease. It's very data-driven, so the possible sources of data that we can use nowadays are clinical data. There's a lot of wearables like Fitbit or other types of devices. We have also molecular data, such as blood tests, DNA tests, and imaging data, such as MRI scans or CT scans. So there's a lot of big data going on in medicine nowadays. And so it's important to make a distinction between big data and data science. The idea of big data is that collecting more data is going to solve your problem, such as, for example, what Facebook is doing with face recognition, where essentially we have millions of examples and we can easily train complicated models. And in many cases, these models are essentially black box models. Another example is ImageNet, which maybe some of you are familiar with. This is a very large database of images that has been used for many deep learning and TensorFlow development, and it essentially contains more than 14 million images that we can use for developing machine learning models. On the other hand, we have data science, where essentially we are more focusing on the problem itself and using domain knowledge. Because in many cases, we don't always have enough data, especially in biomedicine. We don't have these huge millions of data points. And then we essentially want to incorporate information about the domain to be able to model these data. So typically what we have in biomedicine is we have lots of features. We have thousands or even millions of features, so we have high dimensional data, but we typically don't have that many instances. So we need data science approaches, and we need to use deep learning approaches to overcome this lack of annotation. And we need to adapt deep learning to be able to have feasible solutions. So today I'm just going to give you an introduction in how we're using these methods and how we're using deep learning in biomedicine. And first I'm going to briefly introduce what sort of data that we have. Then I'm going to talk to you about some more traditional modeling that was done in biomedicine before deep learning. And then I'm going to give you some cases of how we use deep learning in medical problems. So just a brief overview of what is biomedical data and what are examples of biomedical big data. So one of the things that you might have heard is omics data. This is maybe something that you have heard, the genome, transcriptome, proteome. These are all sources of, these are all molecules that we can measure in high throughput. And then similarly, we also have a lot of imaging data, such as images from pathology or radiology, and we can essentially have all this data of patients and use it for precision medicine and using deep learning. Just a very quick background of molecular biology. So essentially, all the genetic information in your cells is stored into DNA. And that's essentially just like a dictionary. It stores the information. And we can measure DNA. We can measure all the nucleotides, all the ACGTs in each cell of your body. And that essentially gives us millions of data points. Then DNA essentially creates RNA, and RNA is nothing less than the messenger. It's essentially the way that information gets transported. And then essentially RNA forms protein, and the proteins are essentially what is active in the cell, and that's the workhorse of the cell, and that performs all your cellular functions. And all these three levels of molecular biology, we can all measure them, and they have different types of challenges and opportunities. And we can use them to predict diseases, to diagnose patients, and to predict treatments. And this has been possible the last 2 decades because of technology, of course. Initially, there was microarray technology, which is essentially a chip-based technology that is able to measure all these molecules. Then high-throughput sequencing and more advanced technologies were developed. Other technologies, such as mass spectrometry, can be used for protein measurements, and so on, right? So we can essentially get thousands and thousands of features. And sometimes we call this this is essentially molecular data tsunami. We have lots of molecular data, and physicians, doctors cannot really handle this data. They don't know what to do with it. So they need data scientists and machine learning, and they need essentially approaches to find these patterns in the data. So one example of such a large data set is the Cancer Genome Atlas. And this is a project that was funded by the National Institutes of Health. And essentially, it provided information of about 10,000 cancer patients. And it has produced these data for all these different modalities. And also, there are images available for these patients. And you can, this data is all publicly available, and you can access this data. So here is more an overview and an estimate of how much data is available. So there's more than 14,000 patients and over 500 terabytes of raw data that we can use for analysis. And we also have a lot of image data because, of course, for deep learning, images are a very natural way to be modeled with deep learning techniques. And we have 2 very different types of image data in medicine. One of the data sources is cellular images, where essentially we have digital pathology images. And these images look like this. They essentially give us an idea of the cellular hierarchy and the cellular architecture of a disease, for example, a piece of cancer tissue. And typically what happens is a pathologist looks at these images and is manually going to interpret them. But this is very tedious, very time-consuming, and it might also be subjective. If you ask one pathologist to look at such an image and another one, then they might have a different opinion. Similarly, we have also medical images like CT or MR scans, which are maybe more familiar to you. And in this case, what is important to say is that these images are 3D images. So compared to a lot of deep learning that is being done in non-medical images, most images are 2D. But we definitely have 3D images. And when we are using MRI images, we essentially have multi-modal images. So we have multiple sets of 3D images because we can look at different types of properties using these types of scanners. And these images gives us more information about the morphology of, for example, cancers, of tumors, size, location, and so on. And again, there's publicly available data that we can use. So one of the big databases that is available is the Cancer Imaging Archive that essentially provides this data and that we can use for mining. So how are now these types of data, how were they used before we essentially use deep learning? So traditionally, we use more common computer vision techniques to look at these images. And this field is, when it's applied to medical images, it's called radiomics. And it refers essentially to a semi-automatic extraction of features. So we need to look at these images, we define some features that we think are important, and then we extract them from these images. And what these features represent are essentially shapes, textures, intensities, and so on from these images. And there's 2 types of image features that we can extract. And we can extract computational image features and just use algorithms to do this. And in this case, these features are very qualitative. But we also ask radiologists to annotate high-level information about these images. For example, here is an image of a lung tumor. And for properties that we want to extract from these tumors is, for example, what is the edge of the tumor? So the image on the left has a very smooth edge, whereas the image on the right is much more irregular. And so this is just a brief overview of the computational features that we extract. So we look at several texture features and wavelets to see what is the texture inside. Is it very homogeneous or very heterogeneous? We also look at shapes. Is this a very smooth or spherical type of tumor, or is it very irregular? And finally, also, like, we look at the sharpness of the edges. Is the edge of this lesion, is it a very sharp edge, or is it a very blurry edge? So altogether, we can extract hundreds of these types of features. But in most cases, we don't really know what they do. We just plug them into a machine learning model, and we use them for prediction. So here is a brief example. So this is a project that we did where we used data from Stanford patients, and these are lung cancer patients, and we had 85 image features, and we wanted to predict one particular characteristics of these lung cancer patients, which is a mutation in one particular gene. And we used a simple decision tree model to do this, essentially, we were able to predict this molecular property. So using these images, we were able to predict something that's going on the molecular level. And this is this very simple decision tree. So it's essentially just used 4 features to predict this diagnosis. So now, how did we use deep learning, and why are we now using deep learning for modeling these data? So deep learning is essentially a reincarnation of artificial neural networks. And the theory of these models has been introduced already in the 50s, between the 50s and the 70s. But what is new about deep learning is the idea of convolutional neural networks, where you have a convolution operation that I will explain in a few minutes. And these are very powerful techniques to use on text or on images because there's inherent correlation between the features in your data. So what is very important in medicine is that we have growing data sets. We also have lots of different types of data, but the most important point for deep learning in biomedicine is fully automated analysis, is that we can essentially automate these interpretation of maybe the pathology images or medical images. Of course, there's a lot of challenges as well because we have lots of, we have demographic biases in these data sets. We have differences in quality. There's also security and privacy issues in medical data. And also that deep learning models are essentially black box models, so we cannot always interpret them. And this is a very important point for physicians because when we use a machine learning model to help them, they want to also understand why a model comes to a certain decision. So just a brief introduction of convolutional neural networks and deep learning. So essentially this convolution operation, what it does, it's essentially a kernel that is applied onto an image and it can extract certain features from your image, such as it can do blurring, sharpening of the image, or edge detection. So here is a very brief example. So you have this little matrix, which is your kernel, and essentially what you do is you do a sum of products, and this kernel goes over the image and essentially we go from an input image to an output image, and this kernel does some operation on this image. So for example, this is a kernel where your input image and your output image is going to be the same. You can have kernels like this, which essentially detect edges in images. We can have these type of kernels where you sharpen the edges, and then more advanced kernels that do different operations. Of course, we don't know in these medical images what operations are important, so we're going to learn this essentially from the data. So now I'm going to give you 3 examples of deep learning applications in medicine. So the first is how we can use this method for diagnosis of melanoma. So most diagnosis of melanoma is done visually. And automated diagnosis is very challenging because there's a large variety of skin lesions. As you can see here, this is like a picture of all the different types of diseases. There's a lot of benign diseases. There's also malignant diseases, and a physician essentially wants to classify these. Also, this is a very common disease. More than 5 million new cases are being diagnosed every year. and there's about 10,000 deaths to melanoma every year. Here are some example images. So these are just photographic images where the top part are essentially benign lesions and the bottom part are essentially cancers. As you can see, it's not easy. There's a lot of heterogeneity, a lot of variability. How can we distinguish these two types of images? Also, there's different types of images in melanoma. We have smartphone images, which are these photographic images, and from these images, we have a lot of them. And then there's more clinical images, like these dermoscopy images and histological images, but these are more hard to get. So this is the model that was used for this particular project. So they used about 130,000 of these photographic images, and they represent over 2,000 different types of skin diseases. And they essentially compiled over 18 different types of data, 18 different data sets. And then they used the Google Net Inception architecture. So this is already a developed deep learning model that was already pre-trained on about 1.3 million photographic images. And this is a very important point, that we need to do this concept of transfer learning, where essentially these 1.3 million images, these were non-medical images. These were just images of cats and dogs, for example, that were used to pre-train this model. And then we're going to use about 130,000 clinical images to further train this model to a specific medical task. So this is the deep learning model that they used. I'm not going to go into detail here, but you can see they have an input image on the left, all these convolution operations and layers of the network. And then at the end, you have over 700 different diagnoses that the model is essentially going to predict. And this is a visualization of the performance. So this is essentially showing that a deep learning model outperforms dermatologists. So the blue curves are essentially a visualization of the performance of this algorithm and how sensitive and specific it is. And the red dots are individual dermatologists that make predictions. And what this research group at Stanford observed was that we actually can train a model that outperforms these dermatologists. So what are the lessons that we learned from this project is that completely automated diagnosis is really possible, and we can train deep learning models to help physicians. But pre-training is very important because this model was already pre-trained on a lot of other data. And also this medical, this task or this prediction was very similar to a non-medical images, because we have 2D images and we have photographic images, so it's a very similar problem. So now I'm going to go to a more complicated problem, because now we're going to use radiology images to predict lung cancer survival. So essentially we have lung tumors, and we have CT images that essentially give you an idea of the type of the tumor, and we're going to use a deep learning model to predict the prognosis of these patients. This is essentially the question that we're going to ask: is this possible? Can we use a deep learning model just based on images with no input of a radiologist or a physician, and can we use it and can it outperform traditional techniques? So this is the traditional approach, and very similar to the example I gave earlier, where typically what we would do without the deep learning model is we would define some features that characterize these tumors, and these features would essentially give information about the intensity of the image, about the tumor shape, texture, and so on. And then we would have these features, and then we put it in your favorite machine learning model, like a support vector machine or any type of model. And we use that then to predict survival. And people have shown that this is possible. So now what we did is we essentially used a deep learning model. And as input, we use the CT images, and we also use the clinical data, and then we use a convolutional neural network to predict the survival of these patients. What is important in this project is that we have multiple data sets, because we had data from different institutes, because each institute has its own bias. So we need to be sure that when we develop such a model, that if we develop it at Stanford, that we can also use it in the Netherlands, for example. So here are 3 data sets that we use. So we use one Stanford data set, we use a data set from a cancer center in Florida, and we use data set from the Netherlands from the Maastricht clinic. And then we use traditional techniques also to evaluate these models, because we need to train the models on part of the data, and then we test them on data that the model has not seen to prove that we can actually do this task. So one thing that we observed is, so we compare different types of models. We compare models where we only use clinical data, then we use models where we use 2D images, and then we use models where we use 3D images. And here we found that that we really need to use the 3D image information to outperform and to have the best possible performance. So using 3D deep learning was very important in this particular application to predict lung cancer survival. Here's some more results where essentially we tested different types of combinations of training and test sets. So we for example, trained a model on the Stanford data, and then we tested on the data from the Netherlands and data from Florida, and vice versa. Just to make sure that if we train these deep learning models on different types of data, that we can actually generalize to new patient populations. And this is a visualization of essentially the survival of these patients. So that's what we're trying to predict here. And what you can see here are what these curves represent is the probability that a lung cancer patient will die after a period of time, after treatment. And with the picture on the left is essentially where we use both clinical data and the deep learning model, and we found that that model was the best in separating a good group and a poor prognosis group. Whereas if you only use clinical data, which is the picture on the right, we were not able to separate these two groups. So what are the lessons that we learned from this application is that we need data from multiple institutions. Because there's different biases in different populations, different ethnicities that can create different types of response to treatment and how people are going to, what their outcome is going to be. And what we also found was that 3D images are very essential. And a lot of the deep learning development that's going on right now is really focused on 2D images, because in non-medical applications, we see a lot of 2D images. Finally, I'm going to briefly introduce a third application where we're going to look at another type of images, which are MR images. And we're going to essentially, the task that we have here is segmenting these images. So here we have data from a challenge, and this is data that's also publicly available and was provided by the MICI Society, which is the Imaging Informatics Society in medicine. And they have essentially created a data set of over 220 brain tumor samples where we have multiple sets of 3D images that represent different types of properties of these tumors. What we also have is we have this ground truth of these images. So we know from a panel of 11 radiologists, they have essentially classified each voxel in the image and categorized it in different types of regions. What part is the core of the tumor? There's also edema, which are the cells that are essentially spreading in the brain, and they have essentially provided us with this information so we can use it to develop our models. This is another visualization where you can see all these different regions in the brain. So for more details about this, my graduate student, Raghav, is going to give you a very detailed overview of how we developed a deep learning model using TensorFlow to solve this problem. And you can see already some images here about how our model is essentially segmenting these images. So overall, our goals are really to investigate how we can use deep learning and these advances in machine learning to solve problems in biomedicine. And the main idea is really to have automated analysis of these images, because we want to save time for the physicians, and we also want to improve the prediction of diagnosis, prognosis, and therapy response. I hope I've shown you a few examples that transfer learning is very important, because the amount of data that we have in biomedicine is more limited. We don't have millions of data points, and we need large multi-institutional cohorts. The main challenges are actually not technical. So I don't believe that the main challenges are technical, but there's a lot of other challenges in biomedicine. We have issues with security and privacy. Also, as I've said already a few times, the databases are rather small. There's also a lot of heterogeneity between institutions. And there's also more work needed on deep learning for 3D images. These are the people I have to acknowledge for all the work that we do, and thank you for your attention. I'm happy to answer any questions.